So there are loads and loads of antimicrobial drugs that are marketed in the US. There are a vast number of names for all of these. But truth be told, all of them are variants of a small number of drug families. So of about the 260 different antimicrobial drugs that we use, they're classified into about 20 different drug families. We can begin by thinking about them as either broad spectrum or narrow spectrum. Broad spectrum being effective against more than one group of bacteria, and narrow spectrum having a target that's very specific. If we look in this figure, for example, we'll notice that tetracycline has a very broad range. However, penicillin has a much more narrow range. In the rest of this section, we'll explore categories of antibiotics that are based on the 5 major mechanisms that they attack. You probably recall from the previous section that antibacterial drugs target the cell wall, they target protein synthesis, they target folic acid synthesis, they target DNA or RNA, and then finally they could target cell membranes. Let's begin with those that target the cell wall. Penicillins are the primary group here. Penicillin was discovered by Alexander Fleming in the 30s, and since then we've had many more derivatives of the penicillins, all of which end in -cillin. Now penicillins consist of 3 different parts. A thiazolidine ring, which is this yellow ring here, a beta-lactam ring, the red ring, and then a side chain of some sort, which we call an R group. Now, to me this looks much like a house with a detachable porch, and that porch can be a number of different things. Many of these penicillin drugs have been synthesized in the lab, but a lot of them are obtained from microbial fermentation processes. It's a large and diverse group. They all function by blocking the cross-linking of peptidoglycan, as we explored in the last section. However, many bacteria produce penicillinases or beta-lactamases. These are enzymes that are targeted at destroying the beta-lactam ring in these antibiotic structure. So naturally, the bacteria that produce these beta-lactamases are resistant to many of the penicillins. In response, scientists have created some penicillinase-resistant penicillins, like methicillin, nafcillin, and cloxacillin. This table summarizes some of the main penicillin-derived drugs, talks about their spectrum of action, their usages and advantages, and then some of their disadvantages. You don't need to commit any of this to memory, but think about these names all ending in -cillin and being part of the penicillin family. Another family of drugs that target the cell wall are the cephalosporins. They're very similar to the penicillins in that they have the same beta-lactam ring, but they have a different main ring. That's the one in yellow. They have a similar mode of action because they block the cross-linking of peptidoglycans, and mostly their names have the root ceph, as in CEF, CEPH, or KEF. They have a relatively broad spectrum, and they are resistant to penicillinases, so they don't have the same issues that penicillin does. Also, there are fewer allergic reactions in individuals to cephalosporins than we see in penicillins. Now, there are 5 generations of cephalosporins, and they exist based on their antibacterial activity, contain antibiotics that are actually effective against MRSA. There are a few other beta-lactam antibiotics, but these are reserved for use in hospitals, and there are several other drugs that affect the cell wall. However, for the sake of not overwhelming you, I'm going to leave them out in this interest. Before we move on to the next group, let's have a quick concept check. The question is, what is the mechanism of action for penicillin? Does it disrupt protein synthesis, block peptidoglycan cross-linking, disrupt the cell membrane, or inactivate DNA transcription? All of these are mechanisms of which antibiotics attack. But yes, we just covered it. We block peptidoglycan cross-linking. Just checking to see if you're awake. Now we'll move on and look at the antimicrobial drugs that target protein synthesis. This is a fairly large class because, as you'll recall, the prokaryotic ribosomes are different in structure than eukaryotic ribosomes. Protein synthesis occurs on the ribosome, so it's a great target so that the antimicrobial drugs are specific to prokaryotes without doing damage to eukaryotic cells. Again, remember that mitochondria do contain prokaryotic ribosomes, so there can be some effects on the mitochondria. We'll begin by looking at the aminoglycoside drugs. Their structure is of one or more amino sugars plus an aminocyclitol ring, which is a 6-carbon ring. Here we have the 6-carbon ring and then the 2 sugars. Again, there could be one or more amino sugars. The mechanism of action for aminoglycoside drugs is binding to the 30S ribosome. Because so many of them combine to this 30S ribosome, it actually distorts the structure, and that causes error in translations or misreads in the messenger RNA, and thus the protein that's produced is incorrect. These are relatively broad-spectrum drugs because they inhibit protein synthesis. Really, anything that inhibits protein synthesis will have a pretty broad spectrum. Streptomycin is the oldest of the aminoglycoside drugs. It's replaced by newer forms that have much less toxicity. The original form was fairly toxic. Streptomycin is still effective, and it's used in cases where newer forms are not available, like in third world countries. Most of the aminoglycoside drugs end in the suffix mycin. However, this is not really a great way to remember what group they're in because other families use this same ending. Neomycin is another very common aminoglycoside drug. It's used in triple antibiotic creams. Tetracyclines are other drugs that target protein synthesis. They have a 4-ring structure, hence their name, as you can see here. Tetracyclines also work on the ribosome. They block the A site or the arrival site, so transfer RNAs cannot actually enter into this arrival site. If they can't enter into the arrival site, then no protein can be synthesized, even though the messenger RNA has associated with the ribosome. Again, they're broad spectrum because they block protein synthesis. Now, tetracyclines have a few issues because one of the big side effects is gastrointestinal problems. And these gastrointestinal problems are just due to changes in the normal biota of the gut. As tetracycline is having its effect on the unfriendly bacteria, friendly bacteria are being put by the wayside also. So essentially we strip out all of our natural flora and fauna. There are several subgroups and uses for tetracyclines. Doxycycline and minocycline are used for sexually transmitted diseases or Rocky Mountain spotted fever, Lyme disease, or typhus, mycoplasma pneumonia or cholera, leptospirosis, and even acne. Also in this group, we have erythromycin and clindamycin. Erythromycin is a large lactone ring with sugars added. You can see its structure here. It's relatively broad spectrum because it also acts on the ribosome and results in blocking protein synthesis. It has fairly low toxicity, but it's used for things like mycoplasma pneumonia, legionellosis, chlamydia infections, pertussis, as well as diphtheria. Clindamycin is another antibiotic that works on protein synthesis. It's also broad spectrum, but it causes adverse reactions in the GI tract, more severe than most others, so its applications are fairly limited. Synarcid is a relatively new contribution to this antibiotic group. It's used as a combined antibiotic when other drugs are ineffective. It inhibits translation by binding to the 50S ribosome subunit. It's effective against Staphylococcus and Enterococcus species that cause endocarditis and surgical infections, including the resistant strains. It's mostly reserved for use in hospitals. Oxazolidinones are another class. They have a novel mechanism of action because they actually inhibit the initiation of transcription. So as you can see here, when oxazolidinone gets in the way, the large ribosomal subunit cannot join the small ribosomal subunit with the messenger RNA, and thus protein production never even begins. Now, this class of drugs are not found in nature, and the hopes here are that resistance to these will be slow to develop because there's not already a genetic map for the molecules involved. One of the stars of this group is linezolid, or sold as Zyvox. It's used to treat MRSA and vancomycin-resistant enterococcus, or VRE, another highly resistant bacterial strain. Both of these, again, are reserved primarily for use in hospitals. Now we'll look at the drugs that affect folic acid synthesis. These are the sulfonamides, trimethoprim, and sulfones. We've already taken a close look at these, as we have many of the other drugs. Sulfonamides are sulfa drugs. They are the very first modern antimicrobial drug. They're completely synthetic, and they're used to treat things like shigellosis, which is an intestinal infection, acute urinary tract infections, and certain protozoan infections. You'll recall that both of these drugs, sulfonamides and trimethoprim, are involved in this metabolic pathway for the production of folic acid. Sulfonamides take care of this step between PABA and dihydropteroic acid. Don't worry, you don't have to say that one. So sulfonamides inhibit this enzyme, and we cannot move on through the pathway. Trimethoprim will inhibit this enzyme and prevent further movement through the pathway. You recall that they're often used in a synergistic fashion. So trimethoprim inhibits the second enzymatic step. One of the primary treatments for pneumocystis jirovecii, or pneumocystis carnii pneumonia, or PCP, that's found in aged patients, is trimethoprim. Sulfones are yet another class targeting folic acid synthesis. They're chemically related to sulfonamides, however, they have much less broad spectrum effects, and they are key in treating Hansen's disease or leprosy. There are a few antimicrobial drugs that target DNA or RNA, however less, because nucleic acids in prokaryotes and eukaryotes are chemically similar. So those prokaryotes that are infecting us have really similar DNA and RNA. Even so, DNA and RNA have proven to be relatively useful targets for these antimicrobials. Fluoroquinolones are the primary class here. They exhibit several ideal traits. They're high potency and broad spectrum. They address a wide variety of bacteria, gram-positive and gram-negative, and they're readily absorbed from the intestine. There are multiple generations of fluoroquinolones, as there are with many other drugs, but the second generation is most interesting because it includes ciprofloxacin and ofloxacin. Now, ciprofloxacin was the drug of choice in treating anthrax in the 2001 attacks. However, they changed the recommendation to doxycycline, which is used for many other things, and it's a protein synthesis inhibitor. The reason they moved to doxycycline wasn't because Cipro was ineffective, but it's a relatively new drug, and thus it doesn't have much resistance emerging yet. By using doxycycline, we avoid the potential for emergence of ciprofloxacin-resistant strains. So we're reserving that for when we really, really need it. Doxycycline was working just fine. Finally, there's a group of drugs that target cell membranes. But cell membranes are probably the least effective target, as every single cell has a membrane. So the drugs that target cell membranes are not usually the first choice. The polymyxins fall into this category. They have a narrow spectrum, and they're a peptide with a fatty acid component, which allows them to have detergent activity and interrupt the cell membrane structure. The problem with polymyxins is there's a high degree of kidney toxicity, but when it's necessary, they can be used to treat severe urinary tract infections. Daptomycin is another class, and we'll see that daptomycin is actually very successful in biofilm infection treatments. It's a lipopeptide, and it's most effective against gram-positive bacteria. It disrupts several aspects of membrane function. And let's now take a look at how things differ when we get into biofilms. Biofilm inhabitants, as we've already discovered, have different behaviors than their free-living counterparts. And for some reason, when these bacteria are attached to surfaces, they express different genes, which leaves them with different antibiotic susceptibilities. Also, because of the film that's formed, there's penetration issues for many of the antibiotics, so they can't actually get to the cells that are in the biofilm. We also know that cells in biofilm have communication in the form of quorum sensing. And recently, we've discovered that use of aminoglycosides can actually cause the bacteria to form biofilms much more quickly than without the antibiotic present. So we're working on some strategies to manage these biofilm formations, because we see them sometimes forming in the kidney and in the prostate gland, not just on our shower curtains. These strategies involve interruption of the quorum sensing pathways so that they're not behaving differently and communicating to each other. There are some uses of DNase, and DNase really helps with the penetration issues because there's all this extracellular debris, much of which includes DNA from lysed cells in the biofilms. The DNase enzyme seems to help with penetration of the antibiotics through that extracellular debris. But work in the field of managing antibiotics and biofilms is really at the forefront of research at the present time.